on the cell surface or inside the cell. One major limitation is secreted चीजों को नहीं identify कर पाता. And I mean, like the image you see here, there are this is basically like a cell, है ना?
Yeah.
And there are lasers which so cells are basically lined in a sheath. So the diameter of the sheath is very narrow. Only one cell can pass at a time. वैसा रखते हैं, and there is a sheath fluid, and they pass through that sheath under pressure. And as it goes, it is passing through a laser continuously. A laser hits it, and depending on the kind of cell or the kind of antigens that are reacting with antibodies stacked with fluorophores or colored complexes, different light gets scattered. So one is light gets scattered in two directions: forward scatter and side scatter. And then you have something which is fluorescence, which is emitted light. Color is emitted. Now, flow cytometry, when we talk of flow cytometry in our hospital, even it's not in biochemistry, it's there in HMAT. And they use it primarily for immunophenotyping different kinds of cells. The bone marrow is used sometimes to see what kind of clonal cells are there in that mix, right? So suppose you have a bunch of, I mean, suppose it's a leukemia, and you want to know, is it a T cell leukemia or a B cell leukemia? Is it more of CD4, CD8 or other subsets? So the flow helps. Is it myeloma, right? So all those things, the flow gives a good diagnosis. So we'll continue. Flow cytometry is basically a technology rather than, I mean, when you see our previous lectures, this is very different. It's a technique or technology that we're talking of. And when we talk of technologies, we need to understand two, three things. One is the instrumentation part, and second is the application part. And then you have the limitations, advantages, disadvantages, and so on. So essentially, flow cytometry is basically, yeah, as you see, I mean, as you said, it's basically we're looking at cells or antigens or particles which are associated with the cell surface or the cell body. So it cannot identify small molecules which are secreted. So that is one limitation. How it works is it utilizes lasers as a light source, and that light gets scattered, and then it is detected. Depending on the amount of scatter, there is a complex electronic system with software which identifies, depending on how much the light has been scattered, the size of the cell, the granularity or internal complexities of the cells. All those things are identified. And then it can also be tagged with different antibodies, which are, I mean, there's a whole list of fluorophores giving out different colors. And depending on the complexity of the flow cytometer, you will have different multiple channels or filters that are there, and how many lasers are active, right? So based on that, so you may have two lasers, you may have four lasers, you may have four channels, you may have 16 channels. So basically, why it is important is the more lasers and more channels you have, the more number of antigens you can multiplex, right? So by combining different fluorescent probes in a multicolored panel, several parameters can be measured simultaneously. So this is like a step up from your standard ELISA. In ELISA, you have one antigen, one antibody. There's an enzyme-linked immunosorbent assay, and a reaction takes place. Color, or I mean, color is you have a colored complex that is prepared, and based on the intensity of the color, you say, okay, this much was the amount of antigen of interest or antibody of interest. Here, it is similar, but you can multiplex. So especially with things like, say, cytokines, or you want to look at different CD markers at once to identify different cell populations, it helps. The relative abundance of antigens probes in a population of cell is determined by calculating the fluorescence intensity. These signals are then converted to electronic signals, and the file that is created is a FCS data file. Cell populations can be analyzed, or they can also be sorted based on their characteristics. So many times you have flow cytometers with sorters. So you can basically isolate or separate different cells of your interest from a big mixture and then sort them out. Or so the thing is, this is then becomes upstream process for a cell culture or some other downstream research pipelines. Essentially, this is what it looks like. So this is like a multi-parameter flow cytometry in whole blood. I don't want you guys to get overwhelmed, but I would like to draw your attention. You see something here is SSC, like side scatter, and you have something called as FSC. This is forward scatter, right? Now, the final report looks like this. You have a lot of dots. Each dot represents one cell that is there, depending upon the kind of light that is scattered and the degree to which it is scattered. So you will see that more forward scatter and less side scatter. So forward scatter basically denotes the size or dimension of the cell, whereas the side scatter denotes how dense or how much granularity is there inside the cell. So you'll realize that, I mean, when you look at this picture here, you'll see that the granulocytes, the side scatter is maximum, whereas the lymphocytes or these cells, the side scatter is minimum. But the forward scatter, the depending on the dimensions and size of the cells, the monocytes, granulocytes, they're bigger cells. After you have sorted them, before that there are again two more levels of sorting where essentially whoever is doing any experiment, they separate live cells from dead cells, and then they separate singlet cells from multiple cells which are clumped together, and then you go ahead and try to segregate them further as granulocytes, monocytes, lymphocytes. Once you have say segregated the lymphocytes, then you want to know, are they T cells, B cells, NK cells. So over here, suppose you will see that these are all T cells here. The NK cells are over here. The cytotoxic T cells are at this part. B cells are here. Now again, once you have identified the T cells, you further gate them, or you apply different logics where you say, okay, this dimension and this kind of forward scatter or side scatter is accepted, and they are tagged with antibodies, which is giving out these colors. So you have your CD8 and you have CD4. So these letters that you see, they are basically codes for different colors, and I'll show you a list. I mean, there are different shades of red, green, yellow, blue, mix of all that. And based on this fluorescence, they have been able to identify from this whole pool that, okay, these are the CD8 cells. This is the population of CD8 cells. And here you have a population of CD4 plus cells. And again, further, they have tried to segregate T regulatory cells from the CD4 cells. They have tried to gate and see what percentage of cells are the T regulatory cells from this whole mix. And based on that, they identify the numbers and clones and so on. So in this experiment, you'll see there are a lot of things like you have a CD11B, yeah, you have CD14, CD15, CD3, and along with that, you have different names like PECY7, AF750. So these names are codes for different fluorophores that have been used, and they give out different colors. So I have a table later on where you can see the different colors and the names of those fluorophores. So what happens is each particle is analyzed for visible light scatter and one or multiple fluorescence parameters. The visible light scatter is measured in two directions, like we have seen, is forward and side scatter. So the forward scatter indicates the relative size of the cell, and 90-degree in basically indicates the internal complexity or granularity, number of granules, the size of the nucleus, those kind of things. Light scatter is independent of fluorescence because obviously fluorescence happens only when there is a antigen antibody immunoassay reaction that has taken place. And the fluorophores, when you use the word fluorophore, what it essentially means is when it is stimulated with a laser, it gets excited. After it comes back from the excited state, it emits light. So this is how light the fluorescence offers. So traditional applications of flow cytometry involve immunophenotyping of cells prepared from culture, tissue, marrow, or plasma, and those kind of. And they basically recognize specific cell surface or intracellular antigens. Applications now, I mean, there has been a lot of advances in flow cytometry technology. It is now not restricted only to antigens, but can also you can evaluate gene expression, analysis of DNA content, cytokine chemokine production via capture beads, evaluation of cell viability and vitality, monitoring cell proliferation, so on. So, if you remember, we had a lecture previously where we have heard the term called Luminex. Yeah, Luminex is a kind of technology where again it is multiplexing. So, you have to understand the beauty of flow cytometry is multiplexing. One go, you can look at different antigens, you can look at their numbers, their intensity, their positions, and so on. Um, Luminex is one kind of flow cytometry where at one go you have different beads, and different beads are tagged with different antigens or antibodies. And it is so whenever there's a reaction, you have a different signal coming out. So yeah, multiplexing capabilities are immense in terms of flow cytometry, right? This table here shows you all those weird names that we had just seen, like Alexa Floor or AF350, AF405, 488. So you have violet, you have blue, you have yellow, green, right? AF594, PCY7 is yellow, green. So you can see here next to the colors, there are different wavelengths that have been mentioned. And depending on, so when we are multiplexing, we have to be careful, right? We cannot have four different antigens of interest tagged with fluorophores in the same color. You cannot have four, like you cannot use, say, FITC, Alexa Floor, 488, P, and so on, because they're all going to be in the blue green thing. So if you have blue, the laser excitation is blue, but it's emitting a green light. So FITC is green, Alexa Floor 488 gives you a green color, UI light gives you a green color. So if you have three antigens of interest with the same color, you're not going to be able to identify them. So you have to pick one from different emission peaks that are there, right? So that is how you have different permutation combinations. Sometimes, so if you see here, you can have one blue, one green, one yellow, one red, one near infrared, right? So 4, 5 different antigens you can do easy. But when you are talking of multiplexing more and more different antigens, then it becomes difficult, and there is the specificity goes down a bit because you have spillages where you have one detector. I mean, the light being emitted detect and being detected across two different detectors, and you don't know is it one cell type or another cell type. So that is a limitation of fluorochrome based flow cytometry. There are of course different kinds of flow cytometries that we'll see as we go ahead. So when you talk of instrumentation, it basically has three components. It has a fluidic system, an optic system, and a signal processor electronic system. So you see here there is a sheath and it aligns all the cells. The cells pass through in a single line, like a single file. The laser hits the cells. As the laser hits the cells, the light gets scattered. The antigens, antibody reactions that have taken place uh, with the tagged fluorophore, it gets excited and different wavelengths of light are emitted, which is then detected by different, which passes through filters, and there are detectors to detect the kind of light. Based on that, you can identify the cell size, cell granularity, and specific antigens of your interest. And then it is passed through the system there, and there is a software to interpret what kind of light, what it means, the signal, what it means exactly, which cells are more in number, and so on. So over here, you can see, I mean, Many times you have this scatter, and then you can cluster and identify the cells. You have, say, dead cells in this quadrant. If you divide it into four quadrants, you have a left lower, left upper, upper, right lower, and you want to focus maybe on the live cells and the bigger cells or aggregates. You have cells like this, which has less power scatter, meaning they are small, but there is a lot of side scatter. As you go up, the side scatter is increasing. As you go towards the right, the forward scatter is increasing. That means these cells are going to be larger than the cells over here towards the left. As you go up, you will realize that these cells are more granular or more dense. Light doesn't pass through. You have apoptotic condensation of chromatin and those kind of things, where the cells are scattering more light towards the side. So this is how you identify cells, and then you further analyze them in detail, right? So when you look at the peripheral blood, this is what it looks like, a flow cytometry of peripheral blood, whole blood. So you will see here that there are granulocytes. You can see the cells here, granulocytes. There's a lot of side scatter. Yeah. You have your monocytes. The side scatter is lesser than the granulocytes, but more than the lymphocytes. The lymphocytes over here have the least side scatter and also the least forward scatter, because the size of lymphocytes are small, so they are more towards the left and to the bottom. So this is how you interpret the flow cytometric results that are there. How do you link? So flow cytometry allows for quantifying cells and the phenotypes across different parameters. So you have antibody linked to a fluorophore or emission readout. There is a laser which excites it, and you have an emission. So suppose you have cells, you can use cell crushed tissue, you can use plasma, you can use whole blood, depending on, I mean, it needs some cellular content, right? So spleen, dissociate the tissue from dissociate the tissue, homogenize it, make it into a suspension. Then you have to add the antibodies and incubate with a cocktail of antibody fluorophore conjugates. So that's why many times these experiments take longer time. Doesn't happen like our biochemistry reactions just in minutes and hours. Many times you have to keep them for incubation overnight and so on under strict conditions. And then you take the reading the following day. Then the stain sample is made to pass through the sheath. There is laser excitation happening and signals are detected. So this is basically how a flow cytometer works. There may be different permutation combinations uh, in terms of the available technology and sensitivity of the instrument. So depending on what antibodies you have used, you can identify B cells from T cells, from dendritic cells and fibroblasts, because each of them would be having different antigens which are expressed. Right. Now coming to fluorescence. So we saw that there are two things. One is there is an excitation wavelength and an emission wavelength. Right. So fluorescence is basically defined as the emission of light by a molecule that has absorbed light at a shorter wavelength. Fluorochromes or fluorophores are fluorescent molecules, which include proteins or peptides, small organic compounds, synthetic polymers, nanoparticles, etc. Each fluorochrome has a distinct excitation and emission spectra. For example, Alexa Fluor 488 gives you this kind of green color, right? The excitation spectrum is in the range of light that are absorbed by the fluorochrome, and an excitation maximum at 490 nanometers. That's why the name 488. The emission spectrum is a solid green histogram and depicts the normalized fluorescence intensity. So this is basically how the emission happens. And each cell that has been hit or excited, or each antigen antibody reaction gives out an emission. So the software then collates the number of such hits and they call them events, and they identifies how many events have occurred. Because obviously you realize that this is again an indirect method of estimation, and there are chances of false positives coming in, cross reactions, um, cross reaction at the level of antigen antibody reaction. And also because there may be a spillage. It looks green, but green, from our arts class, blue and yellow together makes green, right? So those kind of spillages often happen, and you may get different kind of results from what you expect. Right? So forward scatter, this image is here to give you a better understanding. So light hits it. This is the cell outline of the cell. Yeah, it has nucleus, it has granules inside it. When the laser hits it, it gets deflected by certain angle, and that is the degree of deflection is the scatter. And side scatter is there are detectors at 90 degrees. The amount, so when it hits these granules or the nuclei, the light is again going to get scattered to the sides. So depends. That is why if you have more granular cells or bigger nuclei, the side scatter is going to be more as compared to less granular and anucleatic cells. Right. So forward scatter and side scatter signals generated from the scattering or refraction of laser light. Forward scatter is measured in the forward direction as the laser passes through the cell and is refracted, whereas side scatter is measured at 90 degrees. Both of these parameters provide important information about the cell, appearance, structure, and so on. So this is again the same image you see here. This table highlights forward scatter is for determining cell size. Bigger cells, more scatter, smaller cells, less scatter. And side scatter is for granularity or complexity of cells. Right. Now, after that, it has scattered. If you remember the first diagram, there are different filters that are there. And different filters are made to allow different wavelengths of light to pass. Right. So you have long pass filters which are allowing longer wavelength of light, that is more than 500 nanometers to go through. Short pass filters are allowing only less than 600 nanometer wavelength lights to pass. And band pass, you can decide what wavelength light you want it to pass through the filters, or it will allow only those wavelengths to pass. Right. So, for example, you want 550 nanometer of light to pass with a bandwidth of 50 nanometers. So that means you will have light in the range of 525 to 575, which passes through this bandpass filter. And then they are connected to detectors, and then it is similar to our other analyzers. Anyhow, it has to reconstruct from these signals the kind of cells that are there. Right. Then you have, depending on the number of antibodies you're using or multiplexing, you can have single color flow cytometry labels. I mean, where there's only one antibody, one color, and you're seeing how much of that molecule of interest is present. Right. So you have the cells which get tagged with the fluorophore tagged antibody. There's an antigen antibody reaction. That means you need to give it time. You have to incubate the sample with the antibodies. Sometimes it is overnight. And then after that, you take the reading. When you take the reading, there's a fluorescent signal specifically. And based on that, you identify the type of cell and maybe the quantity or number of cells that are there. Now, after identification, you can do multiplexing using different colors, and then you have, say, I mean, different colors that with different antibodies, and you incubate, you take the reading. This is something, I mean, you will get different fluorescence at different sites, identifying different kinds of cells. Sometimes we want to use multiple antigens for co-localization. We want to see if these two cells are together or nearby. So when you are doing imaging cytometry, this thing helps, right? Again, after that, this is something where there are steps. When you want to identify something, first you identify live cells and separate them from the dead cells, right? Then you identify single cells and multiplex cells. So you have live cells in this area, which is gated. So once they select that specific area, you can program the system to just focus on those specific areas. Then over here, from this, they wanted to focus how much are the lymphocytes. So they are focused on the lymphocytes. From the lymphocytes, they wanted to see between CD3. So you have CD3. The CD3s have separated mode with forward scatter. Then CD3 means T cells, right? Now T cells can be broadly of two types. You have CD4 and CD8. Now CD8 cells and CD4 cells. CD8 cells are bigger, less granular. CD4 positive T cells are more granular and smaller in size. So this is how they have identified. Then again from here, they have identified two different antigens, dextramer 11.6, uh, for staining on the CD8. They've used that, right? And over here, these are the fluorophores that are being used. So these are the fluorophores that they are. So multicolor flow cytometry can be used to create these kind of dots, two parameter dot plots. They're called dot plots. And you have a histogram where the amount of intensity gives us the number of cells that are there. So you can see from this that there are more number of CD8 plus cells than there are CD4 positive cells. Yeah. So you can imagine, I mean, looking at these dot plots, say from the top at 90 degrees, and then when you flip them, you will get these kind of peaks on the histogram. Something the unit they use here is mean fluorescence intensity, MFI, which increases with greater antigen antibody reaction, right? Then you have something known as sorting, fluorescence associated cell sorting. Many of the flow cytometers may or may not have sorters. So here, suppose you want to do further research on one specific cell type from the mixture. So you want a pool which has those kind of cells, right? So from here, maybe you want to focus only on the cytotoxic T cells, the CD8 cytotoxic T lymphocytes and CD4 helper T cells. Now you see there are different cells, different antibodies, different fluorophores, and after they are emitted, you can see okay, all these cells which are there in this region, I want them sorted in a specific event of tube or something else. So when you have these cells, then you can maybe do a cell culture with these cells. You can identify, look at single cell proteomics, genomics, look at all those things in those cells. So what happens is, suppose you have a mixture and you want to look for mutations. The numbers or intensity of those mutations, if they are few, are going to go down because you're amplifying everything, the signal and the noise. With this kind of sorting, you're increasing the signal or your cell of interest, after which you can work on those cells, look at them from, I mean, look at their genetic makeup, look at the kind of metabolites they are releasing, the proteins they are releasing, and so on. So FACS or fluorescence associated cell sorting is one of those things which are used. Yeah. Flow cytometry here is an example where you have to use different gating strategies to identify your cells of interest, and they are multiplexed this for 15 different antigens. And over here they are first gated, I mean CD19 versus CD3. So 19 is in B cells, CD3 is for T cells. So more of 19 is on the y-axis, the x-axis is more for CD3 or T cells. So they have identified this cluster as being actual T cells because there is no CD19 present here. So once they analyze this further with two different and another fluorophore, say for CD8 and CD4. So CD4 is again here, CD8 is this one. Again, they selected this subset, gated it, and then they wanted to see CD8 and CD44. CD44 being activated CD8 or cytotoxic T cells. So the activated ones are going to be to the right as compared to the left. So all this is the CD8 cells, out of which this section is the activated one. Then amongst the CD8, and so they have identified this population for cytotoxic T cells. Now they also want to see how much of the CD4 cells are having Treg cells. So they have compared FOXP3 as an antigen with CD4. These are all helper T cells, and FOXP3 is a known antigen marker for regulatory T cells, Tregs. They are known as Tregs. So in this way, they know that okay, out of all the population, 40.7% are Tregs. Of all the CD4 cells, 40% are Tregs. Similarly, 47% of the cytotoxic T cells are activated. So these are important information that you come to know by gating and identifying these specific subsets of cells, right? So you can see here, CD19 is a marker for B cells, 3 is for T cells, 4 is for CD4 positive T cells. And these are the different fluorophores that they have used. CD44 for activated cytotoxic T cells, and so on. Right. So there is a whole list. These are commonly used ones. I mean, the list is much more extensive than this, but commonly used ones are this to identify your cells of interest. Like CD11B is for macrophage or dendritic. It is expressed by both dendritic and macrophage. But 11C is for dendritic, DC. So if you have CD11B and CD11C, both which are being expressed on a particular cell, then it is going to be a dendritic cell. Whereas you have only CD11B but no CD11C, chances are that it's a macrophage, and so on. Then there can be flow panels to determine the lineages. It's similar to what we have just discussed. And you can multiplex, you can identify immunoglobulins also, IgG, IgM, and so on. What kind of immunoglobulins are being secreted by those cells. So this here is an example of pediatric leukemia, B cell ALL, where you have say okay CD10 versus CD20. Now then again in CD10 versus CD38. So the CD10 is here. You can see here the healthy lineage for B cell phenotype is CD10 intermediate, CD19 positive, CD34 negative, 38 positive, 58 intermediate, and 45 positive. Whereas the tumor or cancerous cells are CD19 positive, CD10 positive, CD34 positive, 38 negative. Because had it become 38 positive, it would have become gone towards myeloma, multiple myeloma. CD58 positive and 45 intermediate. So based on these kind of combinations, you can identify the number of cells which are malignant or which are coming from a particular clone. Similarly, again, it is not just cancer, it can be used to diagnose other diseases also, like immunodeficiency diseases, where you have a subset of cells, because many times immunodeficiency is also at the other end of the spectrum of autoimmune diseases. So it's an immune dysfunction that has taken place. Based on this, you can identify like CD56 positive cells, NK cells are there. Here you have the CD56 versus CD19. So these are B cells. So this is how you identify. Now, there are different types of cytometers that we know of. I mean, there are a lot of subsets, but broadly you have different applications and different purposes and different capabilities of multiplexing for these kind of cytometers. The first type, other than flow cytometry, you have something which is recent and novel, which is known as spectral cytometry. It basically helps us reduce the spillages, and because whenever these uh, fluorophores are used and it is multiplexed, we are going to get a lot of spillage if there are too many antigens you are multiplexed. Spectral cytometry reduces those kind of events and spillages, and you get more specific, sharper peaks which are not overlapping with each other in those histograms, right? Then you have something known as imaging flow cytometry, where again cells are interrogated and images are captured. These based on these images, you identify the size of the cell. They're reconstructed and localized. I mean, different components of the tissue, or even inside a single cell, you can identify what are the structures or antigens of your interest. Where are they located? Are they in the cytoplasm? Are they near the lead nucleus? Are they inside the nucleus? Are they at the cell membrane? Those kind of localizations are possible with imaging flow cytometry. Then recently means in the last 7-8 odd years, there's been a lot of interest with mass cytometry. The rationale being, what if we replace the fluorophores with inner transition metals or heavy metals like lanthanides, right? So in this case, what they have done is there are specific heavy metals or lanthanide group of metals that they have used to tag with the antibodies. So here there is no question of spillage because they are very specific and they're going to give a unique spectra after they're excited, because the properties of transition and inner transition elements of the periodic table they are very unstable. They can be stimulated and they will come back and they will emit light of which are specific for their molecular weights, right? So one more advantage because of this is, I mean, with flow cytometry, you could probably multiplex 8 antigens, 10 antigens, 15 antigens at best with the highest end of flow cytometers. But with mass cytometry, you can multiplex maybe 30 or 40 different antigens in one read, right? So this is one difference. Then this table would help you understand different kinds of flow cytometry techniques that are used, the detection methods, and their applications. Yeah. So this we have already seen. So this is what happens. There is a excitation that happens, and there is a spillover. So suppose there is a value which is happening here. It is being detected. So ideally, it should be detected by this detector between 515 and 545. There's one more detector between 564 and 606, which is detecting these. So this is a false detection at this wavelength, which is happening over there. So this spillover then needs to be corrected or compensated. So spillover spreading is a challenge, and to reduce the spillover is our aim. So that it reduces the resolution of the flow cytometers that is there. This becomes very important as the number of fluorochromes in an experiment increase, that there are higher chances of spillages that are there. Channels that may have spillover spreading issues are identified by creating a spillover spread matrix. So over here, if you look, you'll see that at the top in from detector 2, fluorescence from detector 2 spreads into detector 1, and this spread increases as the signal in detector 2 increases. So if the number of the concentration is more, the spillages are also high. And this is also, I mean, it is logical because the signal intensity of this different antigen may be lower than the signal intensity of this. And this is overshadowing the first signal. That is why if you are not very careful or experienced with flow cytometry, chances are you would miss this signal and you will not identify these subset of cells. You'll focus only on this signal because this is the stronger signal in that mix, right? So the process of correcting for spillover is known as compensation. Compensation ensures that the detector only calculates fluorescence emission specific to that channel. And for compensation, a single stain sample, only one color per sample is needed. These can be set up using cells, compensation beads, capture beads, and so on. And when creating compensation controls, there are four important rules. So you see here, you have a whole bunch. You have the green and the blue and everything mixing together over here, and you can't separate. You have CD25 and CD4. So CD25 is one of the markers which we use routinely even in our labs. It's interleukin 2 receptor agonist CD25. It's a marker for HLH. Yeah, hemophagocytosis, which happens maybe primary or secondary. Often it is associated with immunodeficiency or different cancers. So CD25 is a marker there. Here, it is compensating and it is separating the two CD4 positives from the CD25 positive ones. So how do you do it? You need the compensation must be exact. Fluorochromes in the same channel will still have some spillages. Like we said, when we are choosing the fluorochromes from that table, you cannot have two fluorochromes which are emitting at the same wavelength. Controls must be as bright or brighter than the experiment. So you need to have positive and negative controls which give out a strong signal so that you can compare it with a false signal that is being emitted. And you need enough points or events that are there. So a minimum number of events should be at least 5,000 such points to be conclusive. So these are like rules which are followed when you're doing a flow cytometry based analysis. Right, and also how many events should be collected and those things are there. You don't need a lot of variation. A lower percent CV is always better because that means you have a sharper peak and it's not spread out. So we will not spend much time here. Again, this we have seen you have side scatter, forward scatter. single cells are identified, from them live cells are identified, and then you try to count how many cells are CD8 positive and so on. CD25 positive cells, CD4 positive cells, right? So then you get, when you look at it, you will see that okay, you have CD4 negative, CD25 positive T cells. You have that is represented by the P5 section over here. You have P6, which is CD4 positive, CD25 positive, both are positive T cells. Then you have CD4 positive, but the CD25, so it is on the lower quadrant. So they are the P7 indicating these kind of cells and number of events. So if the events are less than 5,000, it is not very conclusive. The ones more than 5,000 are good, and you are more confident of your results then. So with this, we will summarize that flow cytometry can be used both as a research tool as well as diagnostic tool. It has a lot of applications. There are different kinds of flow cytometry that are used. You have flow cytometry, spectral flow cytometry, imaging flow cytometry, mass cytometry, so on. You can also use flow cytometers for sorting. You have Luminex based platforms where you are multiplexing and doing more and more antigens. Immunophenotyping remains to be the commonest application for flow cytometry, but along with that, you can also do diagnosis for immunodeficiencies, characterize response to treatment, identify and sort different subset of cells to be used in downstream pipelines like cell cultures and so on. You can also do cytochemistry, like you want to look at enzymes, enzyme flow cytometry, where enzymes are being expressed at the surface or in the cell. That can also be done. So that is one angle where biochemistry can use flow cytometry to identify different enzymes, their isoenzymes, their locations, and so on. So with this, I'll end today's talk and be happy to take questions.